So today we have Dr. Alex Fedorec here with us. He is an assistant professor in the Division of Biosciences at University College London, based in the United Kingdom. His research broadly involves applying computational and mathematical techniques to understand and engineer new biological systems. In particular, his work focuses on creating stable and predictable synthetic microbial communities in complex environments, such as the gastrointestinal tract. He is especially interested in biotherapeutic and bioindustrial applications of biological engineering. He had a background in computer science and industrial experience as a software engineer before starting on the path to becoming a synthetic biologist. All right, well, Dr. Fedorec, if you'd like to take it away.
Yeah, thank you so much for the introduction. Let me just try and share my screen. Okay, can you see this okay?
I can see it.
And you can hear me okay? Fantastic. All right, thank you so much, as I said, both to Lee and Alex and all the Gogec team for inviting me to speak, and also for putting on this event. I think it's a really brilliant initiative, and I've been lucky enough to, I was a judge last year, and again, very fortunate to be able to speak to you this year. I'm also very kind of privileged to follow on from Emma's talk. I think it's really great to see what he's doing with Symbio for All Africa. One of my kind of highlights of last year was that I had the opportunity to go to the Symbio Africa conference in Uganda last year, and it was a really an excellent conference, and I met lots of really excellent researchers. And as Emma said, they're throwing their next conference in July this year in Nairobi, so I would encourage people to look it up and try and go along if you have the capacity to. Okay, so as my introduction said, very kindly, my interests are really focused around engineering complex systems. And the complex system that I'm most interested in engineering are microbial communities. So I come from a background in computer science. I really hated biology actually as a teenager, particularly the way it was taught at school. I didn't get on with it at all. But then as a computer science undergrad, I had the opportunity to do a project where I was trying to understand how kind of different social structures arise within kind of groups of animals, right? So we can see things from kind of solitary living animals like a tiger, through to quite complex social structures that don't necessarily map to solitary or social, such as kind of chimpanzees, to incredibly social structures called eusocial structures. In here, there's a picture of these naked mole rats, but I guess the bees are also a classic example. And basically what I was trying to understand is how the kind of the flow of information through the flow of information and interactions with the environment can create these different kind of social structures. And then as I was looking more and more into it, you get to see, well, actually these things happen at vastly different scales across all of biology. So the ones that are easiest for us to observe are, these animal social structures, but actually these happen in kind of unicellular communities of cells. Here we've got a picture of some budding bacteria forming kind of very complex behaviors. They happen within multicellular organisms, within the tissues of developing organisms. There has to be kind of communication across a developing tissue in order to allow it to develop robustly. And even within cells, things, molecular reactions being coordinated within a cell through these complex transcriptional and chemical regulation pathways can produce very complex emergent behavior from very simple sets of rules fundamentally. And so what really interests me is how we can understand these questions. And then as a PhD student, right at the beginning of my PhD, I came across this subject of synthetic biology. And as someone who's, as I say, very naive to biology, it sounded like, well, biology is solved now. All we need to do is start sticking bits and pieces together, and we can create anything that we want to create. Obviously, it's not quite as simple as that, and biology is certainly not solved. But there is still an element of, we know enough now that we can start sticking bits of DNA together and build new biological systems that can perform really quite complex behaviors that we just kind of dream up in our heads. Okay. And one of the things that I quite like to do when I'm trying to come up with a project or an idea to pursue is to try and see if I can fit it into or spread it across these kind of three broad kind of ways of approaching synthetic biology. So the ones that we tend to be most familiar with are these, what I call an application-driven idea of synthetic biology, where we want to perform a particular, we've got some kind of function in mind. So be that producing, a high-value chemical, as is quite common in synthetic biology, engineering organisms to do that, or treating a particular disease. So we've been, as I said, been interested in these biotherapeutic applications of synthetic biology. But application driven is this having in mind that you have kind of a specific goal that you're aimed towards, and that's what you're pursuing. Then there's this kind of exploration driven kind of column of synthetic biology, which I tend to view as the kind of American synthetic biology, because there's quite a lot of research money flowing around in America, and they get the opportunity to play around that many of us unfortunately don't quite get the opportunity to do. But this is the idea that you can kind of dream up something to build without necessarily having an application in mind, just to see if you can do it. And I think this is the area of synthetic biology that tends to be most appealing to me. It feels like, you're a child again, putting together blocks of Lego to see what you can kind of create. And there's some really cool examples of this. What's nice about this exploration-driven approach is that often you may start with no application in mind or no real goal in mind, but it will then feed into kind of research later down the line and prove to be quite important. And then the other angle is kind of a science-driven approach, where we're using synthetic biology to try and answer kind of classical biological questions, from things like how do tissues develop, how do patterns arise, how do particular chemical signaling pathways work. We can use the tools of synthetic biology to answer those questions. And what I quite like to do when coming up with problems to tackle in synthetic biology is actually to try and develop a problem that spans across these three approaches. And hopefully I'll get to demonstrate how we've been able to do that. Okay, so on to this idea of microbial communities. So over the 20 years of doing synthetic biology and the decades of molecular biology before that, we've become really good at engineering, particularly microbes, but also other cell types, to do really quite complex behavior. We can get them to do, digital logic computation. We can get them to produce really complex biochemical pathways and these kinds of things. But there is a fundamental limit to how much we can ask kind of cells to do. They still need to grow. There's this kind of evolutionary pressure on them to grow. And so the more and more we ask them to do, the more likely it is that the systems, the delicate systems that we're introducing into them will break and no longer be functional. So there's long been this idea that we can split these quite complex behaviors that we're trying to engineer into single cells across a community of cells, and that community can perform that function. And in some cases, that community can perform that function better than the kind of single organism can do. This review paper that is pictured on the left, this Johns et al. Paper, I think really nicely summarizes a number of advantages that this idea of moving to microbial communities enables. possibly the thing that I think is actually more important than the benefits of moving to microbial communities in terms of what more we can do is actually that many of the applications of synthetic biology, as soon as we move out of the lab, we're going to be moving our engineered cells into environments where there's already microbes present or other cell types present. And we're going to have to interact or compete or live alongside those microbiomes that are already there. This is kind of particularly obvious when you're talking about things like the gut microbiome, trying to introduce therapeutics into the gut microbiome. Obviously, there's already bacteria there. But even if you want to do something in bioindustry, it's very rare that you're going to have an entirely sterile process. If you want to use cheap feedstocks to do something efficiently, then there's almost certainly going to be some contaminants in there that you're going to have to compete with or deal with. Okay, so really, even if we are engineering single cells to do something cool, we do need to understand that in the real world, they're going to be interacting with and living alongside things that are already present and are better adapted to that environment than the things that we're engineering. So to take that on, so I thought that this audience, being kind of a genetic engineering competition kind of audience, would be reasonably young. So I thought that I'd actually start a little bit with how I pursued an idea through my PhD and how it formed the kind of research that I'm continuing to do now. Okay, so when I joined the lab that I did my PhD in, I didn't really know exactly the thing that I wanted to do. I knew that, as I said, I liked this idea of complex interacting systems and trying to understand how they worked. And then I liked this idea of, well, we've got synthetic biology, so we can build stuff. But I didn't really know what I wanted to do with those two ideas. And within the lab, they were starting to get interested in microbiome engineering and the delivery of therapeutics into the human gut. And one of the problems at the time, although it's less of a problem now with kind of improved techniques, is that when we engineer our bacteria with particular kind of new recombinant proteins, most of the time we do that on plasmids. And then really when you get into it, then you'll stick it into the chromosome. But actually that change between sticking something on a plasmid and sticking something into a chromosome can really affect the kind of the behavior of the circuit that you're working with. So if we can stick with plasmids, then that's potentially a good thing. But to keep plasmids around inside cells, we tend to use antibiotics to make sure that anything that loses the plasmid gets killed off. But when we're working in the human gut where there's a microbiome, we obviously don't want to dose it up with antibiotics to wipe out the native microbiome, because the native microbiome, in most cases, is beneficial to us. Okay, so we don't want to use antibiotics. So we were interested in what other things we could use to try and stabilize plasmids within cells that we're engineering. Okay, and so here's basically a kind of a diagram of the fundamental problem. That when cells divide, they have kind of plasmids within them. And if those plasmids are distributed kind of randomly within the cell, then there's a chance that on division, they will kind of end up kind of spread around nicely, and you get two daughter cells which both have plasmids in. But there's also a chance that all of those plasmids are on one side of the cell during division, and you end up with a plasmid-free daughter cell arising. Now, most of the time, the things that we stick on our plasmids create some kind of a burden within the cell. We want them to produce, a protein of some kind. And therefore, those plasmid-free daughter cells have a kind of a fitness advantage over the ones that are still carrying the plasmid. And so very quickly, we get this problem of competitive exclusion, where the cells still carrying the plasmid grow much slower than the cells, or not even much slower, they grow a bit slower than the cells carrying the plasmid, and very quickly, they get removed from the environment. And so you can do the experiments in the lab, kind of continuously growing these cells, and you see that very quickly, if I bring up the laser pointer, very quickly we get kind of a starting population with all the cells are carrying the plasmid, and very quickly we lose the plasmid bearing population. And so there are a number of different ways that you can try and stabilize plasmids within a population. As I said, the one that we kind of default to in the lab is to stick an antibiotic resistance gene on our plasmids and then just dose up the environment with antibiotics, and that will kill anything that loses the plasmid. But it turns out that there are systems that plasmids have kind of evolved themselves to stabilize themselves. And one of those systems is something called a toxin-antitoxin system. And the way that they work is they produce both a toxin and its kind of antidote on the plasmid. And that antidote is kind of shorter lasting than the toxin. And so when you lose the plasmid, that antidote kind of gets degraded away, but the toxin remains, and it will kill any daughter cells that arise that have no longer got the plasmid. So we tested out a couple of these systems. We were quite lucky that we started with 4 at random, 2 didn't work at all. POXOC at the time, this system was being used by many people, and then reasonably randomly we found this other system which basically seemed to stabilize our plasmids for as long as we could grow. And we were growing for 40 days by this point, and we couldn't get the plasmids to be lost from the system. And this was quite nice. The slightly irritating thing about this for me was that I was doing this work with another PhD student, and he was the one that chose this AXTAC system. And so we had this little bet that whether my system or his system would be better. Turns out that his system, the AXTAC system, was better. But I went through a slightly different approach, right? I had been reading about these things called bacteriocins. And bacteriocins are toxins which are produced by bacteria themselves and secreted into the environment. And they go off and kill anything that is not immune to that bacteriocin. And they tend to target strains of bacteria which are quite closely related to the producing strain. So my assumption had been that given that these were secreted into the environment, they would be able to kind of wipe out anything that that didn't have immunity, whereas the toxin-antitoxin system was kind of a one-shot system, that if the bacteria managed to escape this kind of toxin, then they'd be free to live. But so I lost that bet, the AXTAC won out, and the microcin didn't work quite so well, but it did beat out the kind of the previously kind of go-to toxin-antitoxin system. Okay, so we'd worked out how to stabilize some plasmids within bacteria. And then it really occurred to me that actually we may have now produced a strain that can continue to do what it wants to do, but how are we sure that that bacteria is actually going to stick around in the gut? And how are we going to be sure that not just that it sticks around, but sticks around at a kind of density that we know it needs to be at in order to produce a therapeutic effect? And so this is really where my current work has sprung from, trying to develop this idea of ensuring that strains of bacteria can stick around within quite complex environments, competing with quite complex communities of other cells at kind of known steady levels, even when subjected to noise. And this is really what I see the problem as, right? That we have these kind of potential states within a microbial community. Here we've just got these 2 strains, and we can either have one strain dominating or another strain dominating. And what we might want to do is just have them at kind of this nice even level across time. And that's mostly what we've been able to do so far. But my real goal is to be able to develop a system where we can, in the first instance, control the kind of ratios of bacteria within a community. And then in the long term, get the community to control itself so that it can respond to environmental changes. It can respond to the needs of the community at a particular time, or the goals of the engineer at a particular time, and change its composition over time dynamically without us having to kind of put in any extra stuff. Okay. And this is where those bacteriocins that I lost the bet on actually turned out to be incredibly useful. Okay, so we've been using these bacteriocins as a mechanism for population control. So you can get a strain of bacteria to produce these bacteriocins and secrete them into the environment, and they're able to kill a competitive strain. And so here I've just got a lawn basically of this blue strain, and we drop in the supernatant from this culture that's been growing, producing bacteriocin, and you drop it on and it clears out a zone where those blue strain isn't able to grow. And that basically just shows that we can kill this competitor. Then if we stick it in some liquid culture together, we can grow them together, and you can see that although the blue strain has started significantly higher in this case, the orange strain has been producing this bacteriocin and slowly over time manages to start outcompeting the blue strain. And it turns out that there's a number of things that are really important for the dynamics of this competition. So the first thing is basically just the timing. So you have to give these things enough time to be able to compete with each other. It takes time for the bacteria to produce and secrete these bacteriocins. They have to secrete enough in the environment for the concentration to be high enough to kill the competitor. But also their population needs to be high enough to not be outcompeted at the first step. So here we see kind of time along the x-axis here, and the initial concentration, the initial ratios of these two strains at the beginning. And you can see that when we've got kind of a low number of these things at the beginning, it takes much longer for them to compete with the blue strain than if they start out at quite a high density. And that's pretty intuitive. The more of these killer strains that you have, the easier it is for them to compete with the blue strains. What's also important is not just kind of the relative amounts of these things, but actually the absolute amounts of these things. If you have very few cells to begin with, actually by the time the orange cells have been able to produce enough of this toxin in the environment, basically all of the nutrient has been used up by the blue strain. It's growing faster than the orange strain. It's all been used up, and the orange strain basically has no opportunity to compete with the blue strain. So there needs to be kind of a high enough number of them in the environment in order to compete anyway. So we've got to start with these two kind of dimensions. We've got the ratios of them within the environment and also the absolute numbers of them within the environment. And this produces quite a nice, I don't know if any of you are kind of mathematical at all, but this produces this quite nice bifurcation type plot where we have these two states where in some conditions one of the strains will win out, and in some conditions the other strain will win out. Okay, so here we've got the initial culture density across one axis and the initial ratio of those two strains across the other axis. So in theory, if we can kind of flip between these two states, we can start to change the dynamics of the community in different ways that we want to, okay? One of the ways that we can kind of change these dynamics is actually sticking in a signal to control the production of the bacteriocin into the environment. And here we've used a quorum sensing molecule to control the production of the bacteriocin, the toxin. And we can quite nicely get this kind of dose response like behavior, where the more of the quorum sensing molecule we add, we're turning off the production of the bacteriocin and allowing the blue strain to grow. And it turns out that, yes, if you stick this into liquid culture, then this behaves really nicely, that we can have a system where there's no quorum sensing, the orange strain can kill the blue strain, and when there's maximum quorum sensing, the blue strain outcompetes the orange strain. And if any of you know anything about quorum sensing, you probably know why I chose the quorum sensing molecule. It's because we can engineer one of the strains to produce it themselves, right? And that's going to be really handy later in the next slide. But effectively, again, we can use the data from here to produce these kind of phase-like plots and learn where in space we need to be in order to produce one of the kind of steady state behaviors. So the blue strain winning or the orange strain winning. Okay, so we can link all of this together. And I came up with this kind of scheme in my head, but hopefully I'll show you later that we can do this computationally as well, where we have the orange strain, which is producing the toxin which kills the blue strain, but it also produces this quorum sensing molecule itself, which turns off the production of the toxin. And what this did kind of theoretically in my head, and I was trying to prove that it would do, is that when the orange strain is at a high enough density, it is producing enough of the quorum sensing molecule to turn off the production of the toxin, which means that it's no longer killing the competitive strain. Now, this blue strain naturally grows faster than the orange strain because the orange strain is having to do a bunch of stuff. So when there's none of the toxin in the environment, the blue strain starts to outcompete the orange strain and reduces its amount in the environment. That then reduces the amount of the quorum sensing molecule in the environment, which starts to turn on the toxin. As that toxin turns on, it starts to kill the blue strain, carving out a niche for that orange strain to grow into. So the orange strain population comes back up again. In turn, the quorum sensing molecule density comes back up again and turns off the toxin. And therefore we go round this cycle of the blue strain winning, then the orange strain winning, then the blue strain winning, then the orange strain winning. And so we went about kind of building this full circuit, using kind of a bunch of standard parts that we can piece together. And the key thing here is that we need to really kind of robustly characterize the dynamics of each of these components. So here we've got a component which produces the quorum sensing molecule, and we added in this tuning knob so we can change the rate at which it produces the quorum sensing molecule because we weren't quite sure how much we needed to produce. We can change that by adding a sugar called arabinose. So this module kind of produces the signal, and we can change the amount of signal production using arabinose. The second module kind of senses that quorum sensing molecule and responds by producing a transcription factor. And again, here we characterize its response to the quorum sensing molecule. And then finally, we use that transcription factor to turn off the production of the bacteriocin. So again, when the concentration of the quorum sensing molecule increases, we actually reduce the amount of bacteriocin being produced. Now, by characterizing this system really kind of as robustly as we could, we were able to produce a mathematical model, which allowed us to work out what were the things that we could change within our growth system in order to kind of direct us towards this nice kind of coexisting community that we were after. And it turns out that there were two that were quite easy for us to play around with. The first one was the dilution rate of the environment that these cells were growing in. So these aren't just growing in a batch culture, they're growing in a chemostat. So there's in theory a constant dilution of media in and out of the environment. And that constant dilution not only kind of introduces new nutrients, but it also removes things that have been kind of produced by the community already. So it's washing away bacteriocin and it's washing away cells as well. And then the other key parameter is the production rate of the quorum sensing molecule. And I said that I added in this kind of tuning knob of the arabinose inducible system. And that allows us to slightly tweak the rate at which the quorum sensing molecule is being produced. And with the mathematical model, we were able to see that there is kind of a region in this space where we should expect to see this coexistence of these two strains. And what's better is that actually, to some extent, we could tune the ratio of these two strains within that environment as well. although it turns out that it's a very, very narrow area, so we weren't really able to see that. But anyway, we did the experiment, and my skills at using bioreactors turns out to be absolutely terrible. So I did try growing these in bioreactors a number of times, and I could never quite get it to work. The pumps in the bioreactors didn't seem to like me. So we came up with this kind of pseudo bioreactor type process where you grow your cultures in these microtiter plates, and then you just regularly dilute them either by hand or, if you're lucky, with a kind of a pipetting robot. And that kind of mimics the kind of constant dilution of a bioreactor. And we're able to move basically between these three regions of space, where we've got the blue strain winning, coexistence, or the orange strain winning. We weren't quite able to tune ourselves within this region, because it turns out, as I said, that this region is a bit too small for us to really get into. Okay, so that's a system that we went and built. But this was kind of a design that I just came up with in my head and kind of assumed would work, and was a bit lucky that it ended up working. But ideally, what we want to be able to do is design systems and understand, before having to build them in the lab, if they're likely to work or not. And this is where the computational side of this work really comes in. So what we can do is we can envisage that we have two strains within this community, and they're competing with each other. And those strains, as I've shown you, can produce molecules and secrete them into the environment. Now, the molecules that we've been using so far are these bacteriocins, but in theory, they could be anything. They could be metabolites that could cross-feed with the other strain. They could be other types of molecules that can affect the other strain in other ways. And so those molecules produced by one cell population can influence the growth of the other population. So this can be either positively or negatively. They can also influence the growth of their own population, again, either positively or negatively. And that strain can also produce these signaling molecules. In our case, we've been using quorum sensing molecules, but they could be anything that you could envisage being able to turn on or off the regulation of a gene. And that, as I said, can turn on or off the production of these molecules that are being produced. And those things can all also be done by the other strain in the community, right? So we've already got a reasonably complex web of possible things that we can build into our bacterial community. And how do we know what to go about building? So in this system, with just two strains, one extracellular kind of interactive molecule and one signal molecule, there are 15 different possible ways that we could build this community. And so how do we go about deciding which one to actually build? And the system that we use to do this computationally is something called approximate Bayesian computation. I'm going to give you a quick rundown of how that works in kind of basic terms, but it's quite a neat system. Okay. So what you need for approximate Bayesian computation is basically a model space. So a number of designs that you're interested in understanding which ones are better than the others. And then for each of those models, you can imagine that the production rate of one of the molecules is a parameter. The growth rates of the cells are different parameters. So within each of those models, there's also a parameter space. And some of those parameters you might have been able to fix from experimental data, but quite often we can make design decisions about the rates at which things happen by, for example, changing promoters, changing ribosome binding sites, those kinds of things. And so the parameter space is also can be very broad. Okay. And then we specify some kind of desired behavior for the models that we're interested in observing. So as I've said, most of the time in my lab, we're interested in having stable coexistence of our communities. But we've also explored trying to produce oscillating communities and also chaotic communities. Okay. And so what ABC does, approximate Bayesian computation, what it does is you sample from the model space, basically by picking one of the models at random. And then from that model, you sample from its parameter space. So again, picking parameter values from the possible parameters at random. And then with that model and those parameters, you simulate that model. And then you get some kind of output of the simulation, the behavior of the strains in this case. And you compare that model output to your desired behavior. And if it's far away from your desired behavior, if it looks nothing like the behavior that you want, you discard it. You get rid of it and you say, well, this model with these parameters can't do what I want it to do, so we'll reject it. And then you go on and you pick another model and you pick another set of parameters for that model. You simulate it again, and you then compare the behavior to your desired behavior. And hopefully it will, you do this a number of times and you'll get some that look close to your desired behavior. And in the simplest case, you just really do this at random, right? You do sampling at random, you do your parameter estimation at random, and you just go through, and after thousands or millions of simulations, you eventually get kind of a picture of which models are best at producing your desired behavior. There are kind of more sophisticated ways of doing this selection process, and we use some of them that kind of slowly narrow down the parameter space, the model space, rather than just looking at everything at once. But fundamentally, they all kind of work the same. And what you get out at the end is quite neat, right? So rather than in traditional kind of optimization approaches, where you get kind of the best kind of parameter values for achieving something, here we actually get distributions, what we call posterior distributions, for both the models and the parameters within each of those models. And for the models, what that posterior distribution basically says is whether one model is better than the others and how relatively better it is at producing that behavior. And then we can go in and look at each of those models and look at what the parameter distributions are for that model that are able to produce the behavior. So for some of our models, we'll need kind of very, very specific parameters to achieve this kind of stable coexistence. And for other models, they're kind of much more robust, right? We can have many different potential kind of production rates of a thing. We'll still be able to produce our desired behavior. And those models are the ones that score much more highly, okay? So this is the output of ABC, and it tells us, well, this is probably the one that you want to go and build in the lab. And if you're going to build it in the lab, then you need to know that you need, this production rate to be like this, or this growth rate to be like this. Okay. What's quite neat is that we can actually dig a little bit deeper, because we've got this understanding of how these models look relative to each other. We can start to look for common motifs in these models that are able to produce the behavior that we're interested in. And so here what we've done, we've got this system where we've got 3 strains of bacteria this time, 2 potential quorum sensing molecules, and 3 potential bacteriocins, so toxins. And we've placed some limitations on this, so each of the bacteria can only produce 1 quorum sensing molecule and 1 bacteriocin. So that we had to do to narrow down this space. And still with this system, we've got over 4,000 potential models that we could go and build in the lab. So impossible to know which one you'd want to build in the lab without doing something computational to narrow down the space. It turns out that the vast majority of those models are completely incapable of producing the behavior that we're interested in. But there's nearly 1,000 of them which are able to. And so what we did was we clustered the models that were able to produce something like the behavior that we were interested in. And you should start to see that some of the models are, so the columns here are kind of features of the model. I'll maybe explain that a little bit later. But we did some clustering, and what you should be able to see is that the best performing models are actually quite similar to each other in terms of the motifs that are within them. And then we can look at what those models actually, what those kind of networks actually look like, and compare, perhaps here we've compared in terms of what happens if you kind of add complexity to each of the models. So here is the simplest system that could produce stability in this 3-strain system. And is it worth adding in any extra complexity? In this case, it is worth adding in a little bit of extra complexity to get to the next best model. But probably once you're at this system, there's no point in adding in any extra complexity to the model. And that's really useful to know, if you're trying to build these things in the lab. Adding in an extra reaction, adding in an extra production turn can be really non-trivial, particularly if you have to kind of fine-tune the rates of things. So knowing that you don't have to produce the most complex possible system, you can get away with producing something simpler, is really useful. And if you're particularly eagle-eyed looking at these 4 models that I've shown you here, you might see something that is common between them. And that's that all of the bacteria are producing a toxin which is killing itself, right? So in all of these systems, the bacteria are basically killing themselves. So what we went in to do was look at, well, is this a common motif across the whole of the model space? And it turns out that yes, these kind of self-limiting motifs where you're producing the toxin to kill yourself is much more stabilizing in these communities than if your competitor is producing the toxin to kill you. We don't really understand why, and it doesn't feel like a motif that you'd ever really see in kind of the real world, because you wouldn't expect your goal to be able you wouldn't expect your goal to kill yourself as a population. Your goal normally is to compete with other bacteria. Okay, so that's what we've managed to pull out from this kind of computational approach. And so far, as I've said, we've mostly been working with these bacteriocins and with quorum sensing molecules. And now we're putting a lot of work into trying to create a toolkit which allows us to easily introduce new types of interaction into these systems. So we're really taking this kind of classical synthetic biology approach of modularizing everything that we can, standardizing everything that we can. And so we're trying to introduce things like amino acid overproduction and amino acid autotrophy, so we can do cross-feeding, different types of toxic interactions, internal cellular interactions, so both kind of toxins within the cell which kill themselves. So that's been classically used quite a lot in these kind of self-regulating communities, but also kind of growth genes that we can regulate. And we're also really interested in kind of other types of signaling and communication mechanisms. So trying to move beyond just quorum sensing to, can we communicate more information with the molecules that we're sharing. So we're quite interested in working with kind of horizontal gene transfer, phages to transfer parts of DNA to try and transfer kind of more information in a single signal. Okay, so that's the story that I really wanted to talk to you about today, and I think I'm going to now skip to the end. But I just want to very quickly say that, so this is the area that I've been talking to you about today, this kind of, if we can engineer the interactions between cells within our populations, what can we do? Can we produce these kind of robust coexisting communities? Turns out we can also produce kind of oscillating communities and chaotic communities, as I said. But there's other things beyond just the interactions between the cells themselves. Also, these bacteria, these microbes are within an environment, and that environment can be part of the interacting system. So we from the outside can introduce things into the environment which affect the community, and the cells can also influence what's going on in the community. So we've been doing some interesting things there, particularly using reinforcement learning to understand how we can control the environment in order to control what's going on with the community. And we're also getting into what happens when you add space into the picture. So at the moment, all the work we've been doing is in these kind of well-mixed liquid cultures, ideally kind of idealized bioreactors. But most wild communities are spatially distributed. Right? Within the gut, you've got both kind of the spatial kind of length of the gut, and there's lots of variance in the environment in that length. In soil, you've got kind of things that range from kind of very moist environments to very dry, to very dry environments, very kind of aerobic environments to very anaerobic environments, and that kind of spatial change can really affect things. And so we're also looking at just not just how space affects the kind of the dynamics of the community, but also how we can use things like motility, chemotaxis, as a mechanism to also control the distribution of our microbes within space. And I think I'll leave it there. So thank you very much. I'm going to try and skip to the end of my slides where I've got a little thank you. Yeah, so just to say thank you to the people who have been working on this. So a lot of this work was being done when I was, well, actually I showed you stuff that I did also when I was a PhD student in Chris Barnes's group at UCL. I've got my own group now, and I'm very fortunate to have kind of 3 really lovely and smart PhD students who are working with me on these things. And we've managed to get a little bit of funding, although not at the moment, from various places. So thank you to everyone. Thanks once again for inviting me, and thanks for listening.
Thank you so much, Dr. Fedorec. Would it be okay if we open it up for
Yeah,
questions for
please
just a
do.
few
Absolutely.
minutes? All right, great. Well, if anybody has any questions, feel free to unmute yourself. Okay, Emma, I see that you have a question.
Okay, thank you.
I think you just muted yourself again, Emma.
Okay, thank you so much, Dr. Alex, for that wonderful presentation. My question, I have just 2 questions, and question number 1 is, you talked about the point of coexistence. You were presenting about, okay, when you are presenting, don't you think that at the point of coexistence, if like the conditions change, maybe that point of coexistence, can't we get an issue like maybe one bacteria is eaten up by another bacteria, and we find like now one is taking up the whole system? Then another question is, what is the real world impact of this project, given maybe given okay, let me rephrase it. How can you maybe apply this project in the real world, given of other factors like maybe the environment and ecosystem impact maybe? Yeah.
Yeah, thanks for the questions. Let me start with the second one. So one of the things that I really started getting interested in when doing this kind of community work was, as I said at the beginning, trying to work out how we could introduce engineered bacteria into complex environments such as the human gut. So we did a little bit of work with Tal Danino's group, who developed these kind of cancer therapeutic bacteria, and that's actually where some of that the toxin antitoxin post-segregation killing plasmid stability work was directed. So they now use that toxin antitoxin system in their plasmids for their engineered bacteria targeting tumors. The community control stuff really came out of this idea of, can we engineer a single bacteria to go into the gut and stabilize itself within a very competitive environment, where it's not necessarily adapted to grow there as well as the bacteria that are there. And that's where this idea of this kind of turning on and off competition, turning on the toxin to create a little niche that you can grow into while not trying to wipe out too much of the other kind of existing microbiome came from. So we haven't yet used that system kind of in any animal models, but we are looking at how we can use this to target things like colonic cancer associated bacteria. So there's a couple of bacteria associated with both the onset and poor performance of anti-cancer drugs against colon cancers. So we're looking at how we can introduce these bacteria into those environments. And so those were the main two kind of applications that we've been looking at so far, but we're also interested in their potential for bioindustrial processes. You know, can we use these to control communities that are doing complex kind of production processes? But we're only slowly getting into that area. And then your first question was on, so we're trying to stabilize communities, and how robust are we to kind of other things coming in and potentially eating up our bacteria? I think that was the question. And yeah, so at the moment, the systems that we've designed and built are actually pretty fragile. But I have a PhD student basically working now on understanding how we can make these things more robust. And that computational approach in theory kind of produces designs which are able to be more robust. But also how we can design communities themselves which are robust to kind of invasion and particular types of noise, in particular kind of noisy feedstocks. So again, from this kind of bioindustrial application perspective, if you don't necessarily know exactly the components of the resources that are being fed into the process, can your community adapt to that? I hope that answered the question.
Great.
I guess that answered the question.
Great.
So Alex, may
do you
I?
have a question as well?
So Alex, wonderful talk. Especially the second part is amazing. I have a question for the first part of your talk regarding toxin antitoxin system. You probably understand why I'm interested in that part, because we also develop antibiotic gene-free cloning system. But to develop that, one challenge was, basically when my guess is, you didn't explain in detail, but I guess your system toxin is in the chromosome and antitoxin gene is in the plasmid. Am I right?
No, actually they were both on the plasmid, and both of those toxin antitoxin systems just rely on the difference in the lifespan of the toxin and the antitoxin. So the antitoxin
so stability of the one of the gene, one of the protein.
Exactly.
So in that case, you don't need to rely on chromosome integration of toxin gene.
No.
Then in that case, that's simpler, because of my case is we need to knock out one of the essential gene. In that way, we need to select an essential gene containing plasmid, and then that is one of the selection. Actually, that selection or maintenance, but we also use auxotrophy to select the plasmid containing one. But we now figure out how to do it routinely. But the real question I want to have is, let's say you wanted to develop gigantic, community with a lot of bacteria, and then one challenge might be limiting number of toxin antitoxin pairs for different bacteria. So I just curious how many antitoxin toxin pair you could use practically. There are many top pairs, but practically using those pairs is also limited, I guess. And what is your plan to apply that to real world application?
So for the plasmid, for stabilizing plasmids within each community member, within each population, within a community, actually you can just reuse the same toxin antitoxins, right? Because if it's within a population, then it's not interacting with the other subpopulations within the community.
Right, so but my question is, let's say you're working with the E. coli interaction, that's fine. But if you're talking about E. coli, Pseudomonas, Rhodococcus, you need to find new toxin antitoxin pair in a non-model organism. That is not easy thing to do, I guess.
Yeah, I totally agree, and it's not something that we've looked into yet. No.
But you are going into that direction in the future?
So we're only just starting to branch out beyond E. coli. So we're moving slowly. My feeling is that there will be lots and lots of toxin antitoxin systems that can target virtually basically anything where you get plasmids in those populations. There will be toxin antitoxin systems there, because those plasmids need mechanisms to stabilize themselves. Actually, it may not be toxin antitoxin systems. There's also other types of stabilization mechanisms. But wherever you find plasmids, if you've got bacteria that have plasmids in them, there'll be some kind of stabilization mechanism for them.
Sure. So brilliant. So looking forward to seeing more work in the future. Fantastic. Thank you.
Wonderful. And I think we'll take one last question from Simon, if you'd like to unmute yourself and just go ahead and ask the question. Thank you.
Thank you, Professor Alex, for the wonderful simulation presentation. I did not get something clear on plasmid stability, whereby the second generation of the bacteria, the one which loses a plasmid, having advantages. I didn't get that part, the advantages it gets over the others in the community. And I also like to ask, if this comes into reality, how do you look forward to managing plasmid loss into the environment? Because I presume it can contribute to antimicrobial resistance. How do you think that can be managed in the next step?
Yeah, those are two really great questions. Thanks, Simon. So on the first question, the advantage for losing the plasmid is, so people used to think that just having a plasmid and having to kind of reproduce and replicate that plasmid was enough of a burden to produce this kind of slight fitness disadvantage. And that's possibly true, but that's a very, very small kind of difference in fitness, right? You need to reproduce a reasonably small amount of DNA. That's not too much of a fitness hit. The bigger fitness change is that when we're engineering plasmids, normally the reason that we're engineering them is that we want to stick a protein that is being produced on that plasmid. And so any cells that have that plasmid in them and are producing that protein have some kind of metabolic burden by the very fact that there's resources being taken away from their own growth in order to produce that protein. And so that's enough to create a fitness disadvantage between the cells that have the plasmid and the cells that don't. On the second point about the kind of biosafety, yeah, you're right. It is a potential challenge. There are some really cool systems out there for ensuring that plasmids lost from kind of engineered populations can only grow within particular types of cells. But it's something that in synthetic biology we have to, I think, confront a bit more than we are at the moment, that we're sticking in engineered DNA, and at the moment we just kind of hope that it doesn't get picked up by other things. The hope with the toxin antitoxin stuff is that because we don't need to use antibiotic resistance genes anymore, that at least slightly prevents the spread of antibiotic resistance. But yeah, it's still a challenge.
Great. Thank you so much for all of the questions, and again to Dr. Fedorec for your wonderful talk and your time in Q&A. I believe we're now moving on to poster session A for Gogec, the first poster session, and I'm sure a lot of us are excited to see the projects there.